So here we'll talk about the synapse, and here we're actually starting the third portion of the cells and communication unit. We talked about the cells of the nervous system. We talked about how a neuron generates and conducts the action potential. Now we've reached the point of the synapse, because the purpose of the action potential is to cause the release of neurotransmitters and lead to cellular communication. Now remember, the synapse has three parts: the presynaptic side, the post, and the cleft, the little gap in between. Now, the action potential causes neurotransmitter release. Neurotransmitters are not released one molecule at a time. Rather, they are released via vesicles. A synaptic vesicle is a packet of neurotransmitter. These are made for us by the Golgi apparatus and are absolutely critical for communication. When action potential comes, calcium enters, causes the movement of some of these vesicles towards the presynaptic membrane. These vesicles bind with the presynaptic membrane. The process is illustrated here, and of course, also in the text. These neurotransmitters are dumped into the cleft, and they diffuse over and reach this zone here. This is the postsynaptic density, a zone populated with postsynaptic receptors. These receptors are specialized proteins that can detect the presence of neurotransmitters. We'll talk about what these receptors do, but in essence, they simply control ion channels, much like the ion channels we just went over. But these ion channels, remember, are in the postsynaptic membrane. The ion channels we were talking about were in the axon. Those were voltage gated. We'll talk about these later. But so the process is, you release neurotransmitter. The neurotransmitter swims across, binds with the receptor at the postsynaptic density, causes some effect. Then the neurotransmitter disengages and is cleaned up. Now we have two ways of cleaning up. Apologize. We have two ways of cleaning up. We have reuptake and enzymatic deactivation. Reuptake is the process that I was just illustrating. You have neurotransmitters being released, they come, bind, and they disengage and are taken back up and repackaged. This is reuptake. Enzymatic deactivation is the destruction of neurotransmitters, either by taking them back up or by destroying them. You clean up the cleft so that the next signal that comes across will have a clean plate, so to speak. So we had reuptake, and enzymatic deactivation. Reuptake. Well, guess what? This is our first target of drug action that we can talk about. As many of you know, SSRIs or SNRIs. They largely work very similarly, but these are the more commonly known selective serotonin reuptake inhibitor. These are modern antidepressants. By the way, a better way to think of these might even be, and we'll talk about this later, is as anti-obsessives. Um, that seems to be a better description of what they do, and that's why they work for depression, anxiety, eating disorders, and other compulsive behaviors. Anyhow, what's the effect of blocking reuptake? Well, neurotransmitters stick around and they have a chance to re engage those receptors. So you see, it's a really cheap way to increase synaptic activity without having to do much. In fact, you're doing less. All you're doing is that vacuum protein, you're just deactivating it. That's not very stressful for the cell, which is great. Interestingly enough, enzymatic deactivation goes back a long way in drug discovery. While modern antidepressants work on reuptake, the first generation of antidepressants were MAOIs, monoamine oxidase inhibitor. Monoamine oxidase inhibitor. Now, these monoamines refers to a large group of neurotransmitters. If you inhibit the enzyme that is destroying them, you increase the levels of the neurotransmitter. Believe it or not, and this is very interesting, both the modern antidepressants and the first generations were equally effective. Roughly 60% get better on either treatment. So why don't we use these anymore? These are a blunt instrument. These drugs increase serotonin, dopamine, norepinephrine, epinephrine, and that's too much. The more systems you target, the more side effects you're going to get. These are relatively more specific. These are, slightly more surgical. Okay.